Please welcome Dr. George McGrath, CEO of Lexidus Pharma Services, to present innovation in clinical trials using AI and OCT scans.
Thank you, everyone. My name is George McGrath, and I'm fortunate enough to work for a company called Lexidus. And I'd like to thank, it was an amazing panel to kick this off, and it's just such a pleasure to be up here. You know, at Lexidus, we're a CRO solely focused in ophthalmology with a real rich history in ophthalmology. We've been around for a decade, 180 or so people that get to partner with some of the amazing talent in this room to stand behind you and support you as you develop your drugs and your treatments. And so what I'd like to talk about today is a little bit about what we're doing to try to learn more about our medicines that we're studying. And we're using advanced techniques in imaging to do that. And really, it's I think really critical. It's building on a lot of the work that's already been done. We're just scratching the surface of this, but this is really a passion of mine. You know, folks like Ursula, the team at Moorfields, we work with a group out of Silicon Valley called Voxeleron a lot, and many others around the country have built the foundation of using machine learning and artificial intelligence to understand more about underlying pathology and physiology of retinal disease and that we can't see with the naked eye. And so what we're trying to do at Lexidus is really incorporate this into clinical trials. And I really see the three aims for this, right? So our mission with this, the vision that I have, is that we can use this technology to enroll the right patients that have the best chance of improving with an investigational product, and to learn more about the investigational product along the way. And so that's really what we want to do. And so there's three specific aims, and I'll show you some of the initial data that we have. The culmination of this is likely going to be part of my American Ophthalmologic Society thesis, so it will be published in the next couple years, but we're building out the data set. So the first thing is we want to be able to detect pathology, and today I'm going to show you what we've done for DME, but this is really a platform. We're doing the same thing for dry AMD, we're doing the same thing for inherited retinal diseases, we're doing it across the board. It really is the underlying bones of the machine learning that matter, and then you can point it at any images from any pathology and get a similar result. But for today, we're going to focus on DME. After we can predict DME, really easy, just figure out, you know, whether or not a patient has DME or not without a human involved. The next thing I care about is whether or not we can predict a response to treatment. I'll show you how we're doing that. And then lastly, I want to really be able to take that into clinical trials and try to figure out if we can identify the right patients for the right treatment. So you guys know about diabetic eye disease. It's very, very common, very debilitating. Many of you are doing amazing work on it, whether it's with diabetic retinopathy that some of the group here is doing, or in diabetic macular edema, still an unmet need there, or proliferative disease. And really, the OCT is one methodology, and we are doing work with other modalities as well. But it really gives high-resolution scans through the retina. And so the first question we asked was whether or not a computer can pick out the difference between DME and no DME. It was really a pretty straightforward process, right? So what we did, myself and my partners, these weren't all 6,000 of these images weren't my patients, but I have a couple partners that contributed to this data set. But what we did is we pulled 6,000 OCTs from either DME patients or non-DME patients, okay? And so we went through and we labeled each image as either yes DME or no DME. And then we put it through a computer. And we don't actually know what the computer was really looking at. We just know that the computer was able to predict whether or not it was present, okay? Had roughly the same number of images in each subgroup. And what we found was that the computer could predict 99.6% accuracy whether or not the patient had DME. The way we really did this is we took the 6,000 images, and 70% of them we used to train And then the other 30% we saved and did a prospective study that shows the data here. So really what we proved is in a pretty short order, you can use a computer to tell you whether or not a scan has DME or not. And you can do the same thing with any kind of inclusion criteria you care about. You could put in 325 microns, you can put in whatever other data you want to, but this is just a very simple way of looking at a patient's scan and predicting whether or not they have diabetic macular edema. So the next thing that I care about, and what we're doing right now with the data set, is I want to know if we can predict response to disease. And I think that this is setting up a very nice study, because in diabetic disease, you guys know that the pathobiology of diabetic eye disease is very complex. But in general, in the clinic, we think about two pathways, right? We think about whether or not there's an angiogenic pathway that we can treat with anti-VEGF agents, and we also think about whether or not there's an inflammatory pathway, which we now have multiple treatments, including the newer suprachoroidal steroids that we can use for that. And up until now, our thought process on this has been relatively simple, right? When I was trained how to treat this, I was trained that we start with anti-VEGF because it's safer. And then if a patient doesn't respond to anti-VEGF, we switch over and we'll treat with a steroid. We'll either do IVT or Ozurdex or something like that, or now the suprachoroidal injection. It always struck me, what if we could pick out the patients at the beginning that actually need the steroid, and we could skip the VEGF inhibition? Or conversely, what if we knew a patient wasn't going to respond to a steroid and they just need to keep getting pounded with anti-VEGF, and we spared them from some of the side effects that come with steroids? So the way we're attacking this is we're taking a data set, right? So we're looking at patients before and after a treatment for DME. And you just label these images either no DME or DME. And then you put it through the computer. And the computer should be able to identify the patients that responded to each treatment. And then the computer should be able to differentiate why those patients, or differentiate patients that would respond to VEGF and respond to inflammation. And I think it's a really powerful technology that if we can get it right in sort of this study, and we can use it many other sort of applications. So this is something that's ongoing, and we're labeling the images right now, and we're going through the data set, and we should be able to transfer it to our partners in the computer science department to work with. And my goal one day is to have an algorithm where I can look at an OCT scan at baseline and have the computer tell me whether or not this patient really is a good candidate for a steroid or really may not be, and then use that in my clinical decision making. I would say lastly is that in clinical trials, I think that we are starting to build data sets with early phase clinical trials where we're taking the images and we're running them through the computer, and we're identifying patients who either responded to the medicine or didn't. And the computer is coming up with some amazing insights in areas where you wouldn't expect it. We have one study where really, while everybody's looking at the retina, actually the computer's looking at the choroid, and the thickness of the choroid is a predictor for which patients are going to respond. Something that we hadn't really thought about. So the computer's identifying real trends that are helping us understand medicines that we're studying and giving us more insight into what's actually happening. And I think this is going to grow. A couple days ago, I presented some of the work we're doing at the Dry AMD Summit using other modalities which can be just as powerful, whether it's fundus autofluorescence or other structural tests of the retina. So I think this is a real growing field. It's going to be really exciting to see where this goes. And it's going to be a tool that I hope we can use pretty widely, and I think you guys are going to start seeing data rolled out on this, where it's going to help us really target patients, really make sure that we're treating the right patients with the right drug at the right time. So a little bit about the project team. It was done in academic collaboration, and thank you everybody for the opportunity to present.